whichever you want, except that German is more than rusty. I did my undergrad in Colombia, studying biology, not specializing too much, except my undergrad thesis, I did it on fish. I came to Canada in 94, so a long while back. And I did my master's in Quebec City, Université Laval, where I studied fish biology, cod. In the 90s, there was a lot of money into cod because of the decline. So I benefited of that. And then at the end of my master's, my supervisor offered me a PhD. And I took it on that. And then I went to Louisiana to do a postdoc. And then came back to Sudbury because there was a job. So I'm a fish physiologist. I look into how fish adapt to hypoxia from the physiological point of view. So enzymes, muscle, heart, and all of that stuff. But also, over the last couple of years, I'm a little more into reproduction. So we look a lot into what the capacities of how hypoxia affect the sperm. I focus mainly on males, not so much in females. Whether sperm in fish changes based on stress, environmental stress. And I have been teaching this course for the last 13 years now, I think. So it's a little while. And every year is a surprise because there are so many things that change. So we are going to start with this developmental biology. And my first question is, what is developmental biology? If you already saw the syllabus, then you can tell me a little bit. So what is developmental biology, or what do you expect to learn in this course? Not all at the same time, because the cacophony on my ear is tremendous. Come on, someone. Taya, just because. I know her, so that's why.
Just the development through the stages of life of vertebrates.
Okay, so that's one part of it. Yes, Emma.
More or less like embryonic development between fertilization and birth, or if we're looking at cases where there's eggs hatching.
Okay, yeah, so that is fine, but that is mainly embryology. Okay, when we go through the stages, each stage, and we say, okay, this is the morula, gastrula, blastula, blah, 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 blah. That's embryology. Developmental biology, it talks about everything that needs to be in place for that to occur. Okay, so this is one of my favorite courses for a couple of reasons. This will be the ta-da moment for many of you, where almost everything you learn through these 3-4 years at Laurentian in many courses, you're going to apply it. So we're going to have cell biology, we're going to have molecular biology, we're going to have biochemistry, we're going to have anatomy and physiology, we're going to have vertiform and function, we're going to have a little bit of how environment affects. So is really wrapping up knowledge of genetics too. So little things that you should have learned in many other courses, we want to put it up down here into this course. Okay. And don't worry if you are not good at cell signaling and all that shit. Oops, excuse me. But we're going to go very slowly, okay? So one of the things that I say at the beginning of this course, the first couple of weeks, you're going to say, what the F? Okay? You're going to say, oh my God. And I always say, don't drop it just because it is a little bit overwhelming at the beginning. Okay, so at the beginning, I'm gonna go a little with a whole load of information, but that will be the toolbox. I always call this the toolbox. Okay, so I'm gonna give you all the different things that we're gonna be talking for the rest of the semester. Okay, but then we're going to use them in different ways, okay? So moral for now, don't drop the course after this lecture. Just give me time, okay? For you to be able, okay. So we're going to be talking about a lot of signaling pathways and things like that, but it's the same freaking 4 pathways again and again and again and again to create the liver, to create the eyes, to create the heart, and we see all the interactions, okay? So at the beginning, it want to be a little bit overwhelming, but don't worry. Hey, look at you guys. You don't have exams with me. So it's a lot of work and other stuff, but there is no exams. So no need to learn all these little details by heart, okay? Okay, so I just gonna, let me just grab this. I always need to get into my other screen while I'm doing this. And then you are in one screen and I'm in another screen. And then I don't see you guys. And I'm gonna just stop my video so my little me is not in a corner. But then I can see the screen. Oh, excuse me, I need to share it first, right? Okay, so here we are. So do you see the screen now?
Yes.
Okay, I'm just trying to go to this one. And then I need to move you guys from my other screen into this section so I can see you. Perfect. So developmental biology. So these are the 2 books that we're going to be using. I say the first one is recommended. Okay, so just let me turn my, oh, it's not working, my pointer. I thought that I have charged it. That's what happens when you don't teach for a while. You see my cursor here? Okay, so developmental biology, this is the new book. This is the 12th edition. It's a very great book if you are into developmental biology, if you want to look a little bit on genetics of birth and growth and all that. I highly recommend it just because I followed this book for teaching. So if you miss a concept, it's very easy to follow. Contrary to my other course of vertebrate physiology, where I don't follow one book, but I follow 10 of them. In this case, I do use this book thoroughly, okay? So it's a must if you are the ones that like to read a little bit more and to get more knowledge. This other book is a photographic atlas. This is a mandatory book, okay? Because this is the one that I'm gonna be using for the labs. So I always gonna say, read each section, use this slide and identify the following items in there by following the atlas, okay? So I highly recommend, but this is mandatory because it's all related to this book. So developmental biology, some of you define it as whatever happens from the egg until we have a fetus and is up to birth. So similar to historians, developmental biology established the links among the different events occurring over a lapse of period, that is from fertilization up to birth, okay? So everything that occurs for the formation of an embryo and then the fetus as such, it depends on many different interactions, and that's what we're going to be looking at in this course. So in that end, developmental biology is a multidisciplinary field Okay, it integrates different fields such as embryology, cytology, physiology, anatomy, genetics, and molecular biology. Okay, so this is what I say. This is the Tara moment for many of you, where a lot of things that you learn through the course of the 3 or 4 years, you say, well, when am I going to use that? Well, here it is. Okay, a way to do it. We integrate all of this. Okay. So as such, developmental biology studies all the aspects of development, from genes and molecular events that control the development of the different structures of the body of an organism. Okay, so in this course, we will be studying different developmental stages of animals, mainly vertebrates, although we use the sea urchin as one of the most simple examples. But after that, we move into the rest of vertebrates and the relationship that exists. So even though we like to think that we are superior as humans, we're going to see that from the embryo point of view, we are use exactly the same tools, the same kill tool that a fish use. Okay. So for this course, these are the objectives. We want to review basic concepts of animal development. We want to describe the stages of embryo development in a variety of animal models, and we want to see a little bit of nuances and differences among the different groups. We want to define the principal processes occurring during embryological development: segmentation, gastrulation, ovulation, establishment of access. And each group, we want to see it exactly in this order. We want to see the formation of a sea urchin or a mammal. We're going to follow exactly this. Okay. And we're going to also explain the genetic control when it's present, because it's not always present. And we want to analyze the utility and limitation of gene and manipulation. Okay, so it doesn't seem too much, but we're going to spend the entire semester looking at this. So the structure of this course, because we are in this thing, we want to have 2 courses per week delivered remotely through Zoom, as we are doing right now. During the lectures, we will review basic concepts of developmental biology. You must attend lectures remotely and connect on time, please. You are at home, you don't need to drive. There is not a storm, winter storm outside that make you arrive late. It's just jumping out of bed and plugging those earphones if you have any, and that's it. You can be in PJs, we don't care, okay? So just be on time. And I'm saying this because sometimes if we are watching a video explaining something and you just arrive into the lecture, I won't allow you to go in, because that will stop my video and I need to start all over again. Okay? So just connect on time, please. If need be, because I need to cancel one of our Zoom meetings because I'm sick, I lost my voice, whatever it is, I will create a video covering that material so we don't lose time, and I will upload it as in the lecture section. Okay, where it says lectures, it will be uploaded there. Okay, so I will be doing that so we don't lose any time and we don't get late on our schedule. 1 lab session per week through Zoom. Again, you must connect to all lab sessions schedule. So if you look into the lab section, all the labs are already with the Zoom link associated. So lecture, you want to find it in the lecture, but labs link going to be in the lab link. Going to be 2 right now, 1 at 10 and 1 at 1. Okay, so just choose the correct one. Okay, because it's different. 2% of your final mark will depend on your presence in the labs. Okay. A PDF file of the slides are always provided on D2L. If you look at it, I have already uploaded the PDF for today. Those basically are whatever I have in my screen is what you're going to receive. Okay. At the end of each theme, we will be discussing a scientific article. This discussion will be evaluated. 6% of that final mark is article discussions. So here is the thing. I know that we are in a virtual scenario. Normally, these discussions, we do them in the lab section. Okay, we don't do it in lecture because there is not too much time for it. Okay, so we always have done this in the lab section. So if you read the syllabus, you need to log into perusal.com, create an account. It's free. Okay, I saw this morning that there were 10 students already registered into Perusal. Okay, and you just need to enter this code that is here. Okay, it's the code that is also in the syllabus, and then you get access to my material. So here is the thing. What we're going to do is that I have set the system to create groups. Okay, and you need to read the document, and you need to comment the document, and always relate it to the class. So you do this from your home. You highlight. You have questions. Ask the question, because you can add a little icon with your question. Okay, and if you know the answer, answer to the other person. Okay, so I made this for you to be able to interact with other students in the class. I don't want you to be isolated with no interaction whatsoever with other members, because if we were in person, you will be doing that, right? You will be working in groups, you will be chatting, taking a coffee, going to Tim Hortons and reading a paper, maybe at the pub, who knows, right? So I created this for you to have a platform to interact to one another. Okay, so I have made a setup for 5 people per group, so all of you have the chance to discuss. Okay, and after that, Catherine, your cat is very meow. So cute. And then in the lab, we're gonna do the discussion. Okay, but if you participate a lot into the online, and I see that you have engaged into a lot of interaction with your group of 5, I'm fine with that. Okay, and I have reduced the amount that goes into the class. Okay, but that doesn't mean that you don't need to say anything in the lab section where we are discussing. Okay, so just please register into perusal.com and enter this code. As I say, it's also in the syllabus. We're gonna have a bird effect video. Okay, I am the one that will be assigning the subjects, and I will be haphazardly choosing the teens. Okay, now if you want to be paired with somebody, I need you by tomorrow at the latest to tell me the student that you would like to be paired with. Okay, so just send me that information as soon as you can. If not, I will be haphazardly assigning you with a person. Okay, it's a bird effect. It's a presentation video. Okay, and it's worth 10% of your final mark. We'll see how it goes, because I see this morning I have 55, which is an odd number. Okay, but let's see how it goes.
Excuse me.
Yes.
Just sorry, ignore the noise. We're doing floor downstairs. But for the presentation, would you want us to like talk about it during, like, have a video going in the background and explain it as it goes, or have us explain it?
It's a video that you need to create and put it into Dropbox.
Oh, okay. Okay, so we're not actually like.
No, you are not presenting as we are here.
Okay.
So, and I did that just because a lot of some students may suffer of anxiety, and presenting in front of the class is hard. Mm-hmm. Okay, so I prefer to have a video recording. It's like a YouTube video. Okay. So each of you can present. I do need, however, that the face of each of you appear at the beginning when you present yourself, so I can relate your name, your face with your voice. Okay, it's just, hello, my name is Taylor, or my name is Victoria, and here we go. Okay, so that you can collate that into, there are so many softwares out there, and you are way better than me in technology to do this. Okay, you can record that on Zoom, and then you can put it together. Okay, you can use your cell phone also. There are so many ways that you can do it, and I have a little document that I'm gonna send regarding the creation of the video. Okay. So this presentation will be done. I say at the beginning of the class, but it won't be in class. When I created this PowerPoint, I forgot to change it. As I say, it's need to be done in a video and submitted into Dropbox. Okay. The thing that is true is this is a 5 minutes presentation, and this is not true. So just read the syllabus, because I updated the syllabus 2 days ago. Okay, I forgot to change this presentation. So it's 5 minutes. It's minimum of 4:30, maximum of 5 minutes, and that's it. Okay, there is no question period whatsoever. You need to submit a 500 words abstract of the birth defect at the same time of your submission of the video, and that is worth 2% of that assignment. Okay, and that the 500 words doesn't include reference; it's just the abstract. Okay. There are some indications into how to okay, Annalie, I'm going to answer your question later. Okay. I want you to stick to my directions here. Document in Arial, 12 points, 1.5 line spacing. Margin should be set at 2 centimeters all around. No title page. Okay. And there are some instructions in the syllabus to follow. So it's like, this is the title. These are the 2 students that created the video, and right away. Okay. Pay attention to citations in text, please. I gave more indications in the syllabus. Please follow the instructions. Anything that you write, it will need, has to have, must have a citation. Okay? Unless you did the research and you published the research, then you don't have necessarily to have a citation. But because it's a review document, everything has to have a citation. So the evaluation is video, 10%, term paper is 15%, case study, we want to make 2 in class, is 10%, and there will be a take-home exam case study that is worth 25%. Okay. Participation in lab is 2%. Article discussion is 6%, and lab reports is 32%. So normally for the labs, we never had a lab report for this course. We have just exit quizzes, but because of the virtuality, we want to have lab reports. Okay. So there is some indication in the syllabus. The labs are on Fridays. We're going to release the lab notes a week prior. Okay, so you guys need to read, start developing, answering the questions, labeling the photos that we're going to give you. Okay, you must use the material we give you in terms of the photos. You cannot use a different photo than the one that we have provided. Label those photos and submit by Saturday morning. Okay, so we're going to have the lab session. That's for you. If you have any questions, you can ask those questions at the moment of the lab. Okay, and then you have the rest of the lab time to complete the lab. This is nothing different of what you should have done in a real session, okay, an in-person session, because you get into the lab, you should have already read and answer the questions, and you will have a lab a quiz at the end. Here we don't have that, but you just need to submit your lab report. So you have an entire week to go through, answer, and label. Okay. So the books are going to be using Gilbert, the 2020 version, and Wright for the Atlas. And as I say, for the lab, everything will be related to this Atlas. Okay. I requested to perusal just to see if the lab book can be added into my shelf. I haven't had any responses. However, this book of the Gilbert is now on my shelf. I think if you haven't bought it and you think, you don't need to buy it, okay, through perusal. But you can get access to the online version through perusal, and it's cheaper than the hard copy version, okay? So now let's get into it. All of these 20 minutes. Okay. So the cycle of life. So all animals pass through similar stages of development, such can be generalized, okay? And that's what we call the cycle of life. So stages of development between fertilization and hatching are collectively called embryogenesis, okay? A new life of an individual is initiated by the fusion of the gametic material, so that the male and female genetic material. So now these gametes also are part of this process, the formation. So through the animal kingdom, there are a great variety of embryogenesis, different models, but there is an underlying pattern that includes 5 different stages that are the ones that we have in front of the screen. So fertilization, which refers to the fusion of genetic material from 2 gametes. After that, we have right after the gametes have bound to one another, there is a fertilization, that is the fusion, and then we have cleavage. Cleavage is a series of rapid mitotic divisions that allows for an increase in the volume of the cytoplasm to form a zygote, okay? The zygote is divided into numerous smaller cells, okay? And these cells are called blastomeres, okay? And by the end of cleavage, they form a sphere, like a ball, okay? And that ball is called the blastula. The rate of mitotic divisions starts slowing down, and the blastomeres, all those little cells that I just mentioned prior, undergo dramatic movements. And this series of movements and rearrangements of movements is what we call gastrula, okay? So gastrulation, during gastrulation, an embryo is said to be going through different movements for the formation of the 3 germ layers. What are those 3 germ layers? For those that took histology already, you should know the answer. What are the 3 germ layers? Devin.
Is one of them cellular?
Sorry?
Is one of them cellular?
Yeah, we are made of 3 main germ layers. Ectoderm, mesoderm, and endoderm. Okay, every time that I'm going to talk about germ layers, I will refer to these 3, okay? Ectoderm, the outer layer, mesoderm, the intermediate layer, and endoderm, the inner layer, okay? So after the 3 germ layers are being established, cells going to start interacting to one another. They're going to start rearranging to produce tissues, okay? Oops, sorry. I don't know why my mouse just went off. So we're going to start forming the tissues, and that is what we call organogenesis. Okay, is the cellular interaction to produce tissues, and those tissues then will interact again to form organs. Okay, so we're going to have, for example, the outer layer of the skin is made of mesoderm. The inner layer, ectoderm, excuse me, the inner layer is made of mesoderm. Some cells undergo long migrations to the first place of origin to their final locations. For example, the precursors of the blood cells, okay? The pigment cells, they start always at one point and then they spread very long distances, okay? In many species, and this is not necessarily true for all of the species, we have a fifth stage here, that is metamorphosis, or a larval stage. Okay, the organism hatches or is born, is not sexually mature at that point, so the larva, which looks significantly different from what the adult will become, okay, gonna go through that metamorphosis. So the larvae often have or no capacity for eating, okay, and dispersal stage also, they are not necessarily dispersible, okay. Then in many species, a special portion of the egg cytoplasm give rise to the germ layers, cells, excuse me, and then is the precursor of the gametes, so the egg and the sperm. Non-reproductive cells are called somatic cells. Those somatic cells are the ones that form the body. So the separation of germ cells from the somatic cells is typically one of the first differentiations that occur in the animal development. So even though they're going to be using those germ cells only when they are mature, they are created very, very early in development, okay? So that is one of the first things in development, and we're going to see that when we want to talk about sex determination in the vertebrates. Those cells eventually going to migrate to the gonads, where they're going to find and differentiate, and then development of gametes, which we know is called gametogenesis, is usually not complete until the organism has become physically mature. So this figure illustrates the developmental history of the leopard frog, Rana pipiens. So we start here with the joining of the genetic material of the sperm and the egg. We have the fertilization. We want to see all that. So this is very quick, okay, at least for this frog. Then we have the section of cleavage that I was saying, that is the highly rapid cell division of the cytoplasm of the egg, okay, until we get, so we have the morula and then the blastula. It's called blastula because you have a blastocele. Blastocele is this little hole inside, okay? We're gonna be talking about this in detail later. Then you're going to go into gastrulation. Gastrulation is the transformation and migration of cells to become a tryphoblastic embryo, which have the three germ layers: ectoderm, mesoderm, and endoderm. So here we see how it's moving, all of this, until we get the ecto, meso, and endoderm. And then we're going to go into neurulation. That is part of the organogenesis. So in here, the three germ layers start interacting in order to form the different parts of the embryo. So all of that encompasses organogenesis. And then, as I say, they're going to hatch or they're going to be born. And some will have the larval stage, that is in the case of the ranapipiens, until then it gets transformed into maturity, and the gonads that we see here start forming, migrating until they become mature. Any questions up to here? No? Okay. So gametogenesis and fertilization is, in most frogs, gametogenesis and fertilization are seasonal. So we have seasonal cues that allow us to know that is the mating season. Increasing hormones stimulate the production of estrogen to increase the size of the ovaries. The yolk start migrating to the ventral hemisphere. The upper half is called the animal hemisphere. So we have two sections. We have the animal part and the vegetable part of the egg. Okay, so that's normal formation. We have a lot of pigmentation in the animal hemisphere in here that is made of melanin that protects the egg against the UV light. Okay, in the vegetal, excuse me, hemisphere, we're going to find a lot of the yolk. Okay, the sperm gets into the egg. We're going to have a lot of reactions that we want to see later that causes a movement of the yolk. So right here we have an activation, okay, and to create the gray crescent. We're going to talk about this gray crescent at length because it has a lot of information that will be required for the egg to become active. Okay, so in here we have the female pronucleus. I'm going to have the male pronucleus. They're going to bind to one another and they're going to fuse. And once they fuse together, the cytoplasma is when it does this movement. Okay, so the cytoplasm moves to a new locations determined by the three body axes in here, which is absolutely important. So there's section for the enter and the opposite start moving, but don't worry, we want to talk about it later, okay? So finally, all of these events that happen just by the entrance of this sperm allow to determine which part of this egg will be the head, the tail, bottom, top, right, and left, just by the entrance of that sperm, okay? So it's a lot of things that are going to happen in here. Cleavage and gastrulation. So during cleavage, the volume of the egg does not change. Okay, I just want to make that point. The volume as a whole doesn't change, but it is divided into hundreds of thousands of cells. Okay, so in here we see the egg that just was fertilized, and look at that, it hadn't changed the shape, the size, right? But it has way more cells than just the one that initiated it, okay? So the sequential divisions are not synchronized, and the cells at the animal hemisphere divide faster than the ones that are in the vegetal hemisphere, okay? And this is just because of the vitellus that is in here. Those big goblets of fat and proteins and so on slow down the division, and the division is faster in the animal pole of this new zygote, okay? So as they divide, they create a blastocele. So this is the blastocele that I mentioned. It's a cavity, okay? And this cavity, this blastocele, always is placed in the animal hemisphere or in the animal pole, okay? The blastocele is very important during the process that follows, that is gastrulation, because that is what allows those layers to place in the right position, okay? So gastrula, which, as I say, is the migration of the cells. So we have all the cells divided here at top, and then here by these arrows, we see that these cells are migrating on each side, okay? Until they get into the very end, and we only see the yolk plug, okay? So we're gonna see all the different movements that occurs to be able to create this trophoblastic embryo. Okay, so we see that the blastocell, as the cells start going through epithelium, we're going to define those terms in a couple of minutes. Then we have the ectoderm and endoderm, and here where we saw the gray crescent, that point is going to become the dorsal lip of the blastopore. This is a signaling section that has so many factors that allow the formation and movements of those cells. And here we see that the first cells that come here start getting in, and look at that. We have now the creation of a second cavity that is called the Archenteron. Okay, this is the blastocell that started here, but as these cells start to invaginate, then we have the Archenteron, and the Archenteron going to play an important role also here. Okay, until we get the ectoderm, mesoderm, and endoderm. So just be aware that from now on, these colors going to be seen in the same way in every lecture and in the lab. Ectoderm always blue, mesoderm always red, and endoderm always in yellow. Okay. So now the next step is the morula. So now we have cells all around, right? So we're going to have here a lot of cells, and that's when we say that that embryo is called a morula. Okay, so we're going to have organogenesis going to start at this point. So the beginning of the formation of the notochord that has been signaled by the interaction of the placement of these 3 germ layers. But we're going to see that in detail later. Okay, right now it's just an overview of the stuff. Okay, so in morula, so we have here our Archenteron, right? That it was the one that we saw prior. Okay, so in here, this Archenteron is going to play a very important role because it's in right now it's in the middle, and then we're going to have the formation of the neural tube. Don't worry, we're going to have a full lecture of the neurulation and formation of the neural tube later on. Okay, but here we see the neural plate that is ectoderm, then we have the mesoderm, and we have the endoderm. And we're going to see how these edges going to start rising, folding, joining, and start from the head. And then we have here the back of the embryo. And this that we call the blastopore. Okay, so we have a cranial blastopore and the tail blastopore in here. And every part of this going to have different molecules, different signaling pathways that are going to be followed to determine this is the head, and these are the structures that are required in the head. This is the trunk, and these are the parts that are required at this point. Okay, so we're going to see all of this in detail through the course. So starting by defining the embryo, then we say forming an embryo is the hardest thing you will ever have to do. This is a quote from Gilbert. As an embryo, you had to build yourself from a single cell. You had to respire before you had lungs, digest before you had a gut, build bones before when you were all pulpy, and from orderly array of neurons before you knew how to think. So it's a miracle, really, that we are right now sitting here. Yeah, we all have our aches and pains. We have some illnesses and things like that, but we're alive. We could have failed in the formation, right? So one of the critical differences between you and a machine is that a machine is never required to function until after it's completely built. So every animal has to function before it is built, and that's when a lot of things can go wrong. So before we can answer how did we become what we are, we need to understand how everything is interconnected and plays a very well orchestrated symphony to create the beautiful beings that we are, or the beautiful cat that we have on the side, or the beautiful Chico. Chico is my dog and he is behind me and he snores sometimes and you can hear him. And sometimes he just loses it and starts barking. So I apologize on his behalf from now on because it does happen that he does that. So to create all organisms is like a really good symphony, right? Every instrument needs to be in place at the right time. If it comes late, if it comes too soon, maybe some defaults gonna occur. And this is why we have some birth defects that we're gonna be looking into this class. So several fields can be used to understand what can affect embryos and allow their development. So we have comparative embryology, so that allow us to see how anatomy changes during development in different organisms and how comparable we are at the very beginning of development. We have evolutionary biology that allow us to see changes in development cause evolutionary change. And we know that some individuals have lost some wisdom teeth. Some individuals have lost a finger, right? Even though we all came from the same group. And also teratology, that is the study of birth defects, and that every chapter we want to see something that can go wrong, okay? Congenital malformations also. And this is very, very important field nowadays because we are so exposed to so many teratogenic substances every day. So a little bit of history, very short. So Aristotle was a Greek philosopher and scientist and considered as a pioneer of embryology and developmental biology. He was the first study to compare different ways how animals are born. So he was the one that determined oviparity, that is, individuals that are raised from eggs. Most invertebrate frogs and birds are, say, oviparous. Viviparity are the placental mammals. Okay, so we have placenta and birth. And then we have ovoviviparity, like sharks, for instance, and some reptiles too. Okay. Aristotle also defines cell division patterns, okay, by which embryos are formed in two different ways. So we have holoblastic, which means that the entire egg divides. And then we have meroblastic, which means that only a part of the egg is destined to become the embryo, while the other portion, which is the yolk, serve as a nutrition. And this is the case of chicks, for instance. They have meroblastic cell divisions, okay? And the holoblastic, we have what? We have frogs and we have mammals, okay? After Aristotle, there was very little progress in embryology until William Harvey in 1651. He negated the idea of a spontaneous generation of animals from mud and excrement. That was against the views of Aristotle, because he said that, in the mud there are plenty of things that grows. So for him, that was the spontaneous generation. And embryology was mostly based on speculation before the invention of the microscope. So that is when Malpighi in 1672, with the creation of the microscopy, was the first one to be able to observe the chick development. And that's what we have here in one of the uses of the microscope that he did. So that's a 2-day-old embryo chick made by Malpighi. So there are different patterns of embryonic cleavage, as we just mentioned. And so different organisms undergo cleavage in distinct and different ways. Pattern of cleavage is determined by two things: the amount of yolk and the angle of the mitotic spindle. Okay? If one pole of the egg is relatively yolk-free, cleavage occurs faster okay? Because the yolk normally has the tendency to inhibit cleavage, or at least to slow down the cleavage, okay? So normally the vegetal pole is the one that is rich in yolk. Here in this image is at the bottom. This is the sea urchin and this is the frog. So this is all the yolk, so is the vegetal hemisphere. And the animal pole or animal hemisphere normally is at top. But if you flip the egg, well, you have it upside down, right? So it's better just to say that the animal pole is the section that is yolk poor and where mitotic division occurs very quickly. And the vegetal pole is the one that is rich in yolk and normally slow down the division, okay? The zygote nucleus is usually displaced normally towards the animal pole. So this figure is in the book and illustrate the classification of cleavage types and the influence of yolk on cleavage, okay? So holoblastic, we say that the holoblastic, we have the entire cytoplasm divides, right? So that's at one extreme, okay? So at one extreme of the egg of the sea urchin, this is the one that we're going to be looking at, the snails and mammals, their eggs have a sparse quantity among the yolk, okay? And that what we call isoleucithal, okay? So a sparse, evenly distributed yolk. So the yolk is evenly distributed along inside the egg, okay? Now, in echinoderms and Amphioxus, we have a radial cleavage. So we have 1, 2, and then equatorial. Okay, meridional, and equatorial. Then we have a spiral cleavage in some worms that it goes like in a spiral. It takes the name. Then we have the bilateral cleavage in tunicates. Okay, and then the rotational cleavage. So holoblastic, complete cytoplasm divides depending on the angle that division of cleavage occurs. It takes different names, okay? So little yolk, evenly distributed, and the whole egg divides. The second type is the mesolacital. Mesolacital means moderate amount of yolk, mostly on the vegetal yolk, okay, vegetal section. So placed essentially more towards the vegetal pole, and normally it's a displaced radial cleavage, okay? So we have meridional, and then we have one that is displaced here, okay? It's not at the equator. Equator will be a little more here, right in the middle. So that what is displaced, okay? At the other extreme are the eggs of insect, fish, and birds. So we have a bigger size of eggs, okay? And most of their cell volumes are made up of yolk. So the yolk must be sufficient to nourish the animal through the embryonic development. Zygotes containing large amounts of yolk undergo always a meroblastic division, which means incomplete cleavage. So only a section will be divided. So the cleavage furrows, which are these ones, you see, are just like furrows. It just go a little inside, but doesn't go all the way down, okay? It's only arriving at the animal hemisphere, okay? Not in the vegetal, but in the animal. So in here we have cephalopods. Molluscs are bilateral cleavage. It's all there. This coidal cleavage is what we're going to see in class. It's going to be fish, reptiles, and birds. So we have a blastodisc, okay? And it's only the cells in the blastodisc, the ones that are gonna divide. The rest don't divide at all, okay? And those are, say, telolecithal, because it has a lot of yolk, okay? Now we have the centrolecithal. The yolk are in the center of the egg, okay? And this is most insects has this one. So we have a superficial cleavage division, and the cytoplasma in here occur only at the periphery of the cells, but the rest is empty. So start here, and then start going to the sides. So meroblastic and divided in telolecithal and centrolecithal. Okay, gastrulation. I'm just looking at the time, it's 11. Okay. Gastrulation is a highly coordinated cell and tissue movement where the cells of the blastula are dramatically rearranged. Okay. During gastrulation, the cells of the blastula are rearranged and are multiplied in multi-layers on the organism, and that's what allows the establishment of the gastrula. So the germ layers are established. Future mesoderm and endoderm cells move to the interior, and the ectoderm cells will form the skin and nervous system. Okay, so it's gonna go all the way around and then gonna cover. Okay, the stage is set for interaction between the newly positioned germ layers. So here we have our blastula. Remember, this is the blastocyst, so that's what we know is a blastula. And then we have the animal pole. We know that is the animal pole in here just because it's dark, where all the melanin was to protect the cells. And in here we have our gastrula. And we know that it's gastrula because we see the remnant of the blastocyst, right? And then we have the Archenteron that forms. So we have two cavities here. So that's what we know also is a gastrula. So this is a table that mentions all the different movements. So during gastrulation, I say there are different movements of different cells. So, and the first one is invagination. It's like you take a balloon and you put your finger inside, right? In the balloon. So it's an enfolding of the epithelium of cells. And here, so it goes in, invagination. We see this in sea urchin for the formation of the endoderm. So as soon as it gets in, now it becomes endoderm, which means it has another set of signals, okay? Then we have involution. Involution is like a rolling over of one layer of cells, and it does it over a lip, what we call a lip. So this will be a lip. So these cells were here, went all the way around, and then at the lip section, they start rolling inside, okay? So it's an inward movement. And we see here an example is the mesoderm of amphibians formed in this way. Then we have ingression. Ingression is the migration of individual cells from the surface layer to the interior of the embryo. So they lose connectivity in this section and they start migrating inside. And this is how the mesoderm of the sea urchin is formed. So individual cells lose connectivity and then go. Then we have delamination. Delamination is simply the splitting or migration of one cellular layer into two. So here we have one layer that divides, and now we have two. That's delamination, okay? And one of the examples is the formation of the hypoblast in birds and mammals. We want to see that in detail. And then epiboling, which is one of the most fascinating movements, is like the movement of the epithelial layer, which is the ectoderm in blue, right? And spreading, covering. It's like we have a balloon or something, and when then, or a strawberry, right? And we put all the chocolate on top and covers the entire thing. Decadent. That's the movement. So the ectoderms are going all around the ball of the cell and covers. And that's how the ectoderm formation in all of the different animals occur. Okay, so invagination, involution, ingression, delamination, and epithelial. 5 key movements. Please learn the terms, because from now on, I'm going to say this forms by delamination, this from epithelial. Okay, so just remember these figures and what they mean. Axis formation of bilateral symmetrical animals. Even though it says bilateral symmetrical, we are not that symmetrical, right? We have an eye that is a little smaller than the other, an ear that is, and if we look at each other, well, we are far from being symmetrical. So the development of embryonic axis is really critical. And there are 3 critical axes that must form in every embryo. One is the anterior posterior. Where is the head? Where is the tail? Okay. Where is the mouth? Where is the anus? Very important, and require different signals to know which part are you in and what are you supposed to form. Then we have the dorsal ventral. Okay. Where is the back? Where is the belly of the individual? And then we have the lateral, which is the right and which is the left. Okay, so anterior, posterior, dorsal, ventral, and lateral. With all this, we also have different planes of view. And this is becoming very critical for you guys in the lab. Okay. Because we have mid-sagittal is in the middle. Transverse plane is divided section in anterior posterior, right? It's a cross section. And then we have the horizontal plane that divides into top and bottom. Okay, so keep in mind these ones because you're going to need them in the lab. Okay. Now we need to name the parts that are going to be forming in the embryo. So with the new technologies, we have so many things. We have improved microscopes. We have stains, life stains. We have so many different techniques that allow us to really follow the formation of each part of the individuals. So descriptive embryology evolves so rapidly. And there are 3 main scientists in this field. And we have Ponder. Okay. Ponder determined the germ layers. Ectoderm generates the outer layer of the embryo, epidermis of the skin, brain. Blood, heart, kidney, gonad, bones, muscles, and any other connective tissue that we have in our body. Okay. So that was Ponder, the first one that described these 3 germ layers and what they give rise to. In here, he also defined the pharyngeal arches that we see here in this figure. Okay. And we see the pharyngeal arches, we see them in all groups, even in humans, okay, being part of the inner ear. So that's what we see in this other figure, okay. So in here we have a fish, reptile, and a mammal, fish, reptile, and the mammal, and we see how the reptilian, the gill arches, the reptilian jaw articulation bones, and the mammalian middle ear bones are all part of this same homologous structure. Okay, so here we have the iomandibular bone in reptiles, amphibians, and birds, the posterior portion of this cartilage from the quadrate bone. Okay, so here you can see it here. And in the lower is the articular. This is in here. And in mammals, this articulation migrates to a different region. That is the dentary and squamosal bones that are both in this section. So, but all came from this same structure. So in this case, we call them homologous. It has a same origin, but then they're placed in different and accomplish different functions. So Carl Ernst Bombaur continued panther studies and noted that it's a common pattern to all vertebrates developing. He discovered that each of the 3 germ layers generally gives rise to the same type of organs. He discovered the notochord, the mesoderm axis that depends on the left and right side of the embryo. That is so important in development. If we look at these 2 embryos, one is a lizard, one is a human. Could you say, really looking at it, oh no, this is a human? Not really. They are almost identical, right? So each embryo begins with the same basic features, although they acquire structures at different ages and sizes. So from the embryological point of view, they are very, very, very similar, okay? So from the observations, Carl Bombaur postulated 4 different principles that are the ones that we see here in the screen. He says that general features of large group of animals appear earlier in development than specialized features in small groups. Less general characters develop from more general until finally more specialized characters appear. Okay. So a lot of differences between the leg, the wings, and the arms become apparent later on development. At the beginning, we don't see too much of the difference, but it becomes later. That's exactly what that second point wants to say, okay? Embryo of a species, instead of passing through adult stages of lower animals, departs more and more from them. So all of this, what I want to say is that before, there were some assumptions that the embryo was like a mini-me from a start point instead of going through development, okay? If that is true, therefore, early embryo of high animals is never like adult of a lower animal, but only like its early embryo. Okay, human embryos never pass through a stage that looks like an adult fish or a bird, right? Rather, a human embryo initially shares characteristics common with fish and birds, but it's not necessarily the same. So, in here, we have that we can follow what is going to occur with some cells very, very, very early during development. Just give me a second, I need a sip of coffee. It's cold now. Anyways, so normally we say that a tissue is a tight junctions of different cells that are connected. So cells, mesenchymal cells, gonna lose connectivity. Sorry, the buzzing is my dentist is calling. So these cells, mesenchymal cells, gonna lose connectivity and they become mesenchymal cells. And this is the epithelial mesenchymal cell transition. And this is what we see here. So these cells are capable of going into a different arrangements of cells. Okay, so there are 2 major types of cells making the embryo, right? We have the epithelial cells, which are tightly connected. That's what we have here. Okay, or sheets or tubes, but we also have the mesenchymal cells, and that mesenchymal cells are loose, they don't have any connection, and act as as an independent units. So between epithelial cells and mesenchymal cells, morphogenesis can use a limited repertoire of variation that it possesses. So here we have some of those things that they can do. Direction and number of cell division, okay? So we know that animals come with different sizes, which means that some will have more cell divisions than others, okay? For instance, look at the size of the head of each of us on the screen, okay? We have the small heads and we have bigger heads, okay? Which means that one has a little bit more of cells than others, okay? And cell shape changes. What it means, this is a critical point in development. We need that cells change their shape depending on what they are creating. They are creating tubes or they are creating layers. The shape is different, right? Cell migration. So cells have to move in order to get to their final destination. Just give me a second. Le téléphone. Okay. My husband also is working and he has his headphones, he doesn't hear. So cells need to move in order to arrive to the section that they want to form, okay? Then we have cell growth. We don't think too much about this, but cells like germ cells, for instance, change their size. For instance, a sperm eliminate most of the cytoplasm that they have, right? And they only have the head, the neck, and the tail, okay? As gonna be seen later in the class, some cells placed at a certain section of the embryo will be smaller, while others will be bigger. And then we have the last one, but not the least, cell death. Death is a very important process of life, okay? At the embryonic level, it allows, for instance, the creation of independent fingers. Imagine if we didn't lose by programmed death the interdigital membrane. We all have palmated hands at the beginning, but because of the system, we're going to see that one of the last chapters that we're going to see, the tetrapod limb, we lose that membrane, okay? Chickens and ducks have them too, except that in chicken is lost and ducks keep it, okay? So we have apoptosis program cell death that is important. The creation of hollow structures in our body, like the vagina, like the mouth, also goes through the process of death to open up that cavity, okay? So we can follow the origin and final destination of cells using a fate mapping of the embryo. So we're going to trace the cell lineages, okay, which normally is one of the most important programs for descriptive embryologies because they're going to follow individual cells to see what they become, okay? We have fate maps. The fate maps are simply diagrams like the one that we see in here in the screen of the larvae and adults, and we see the region of the embryo from which some structures arose, okay? So in here we have the zebra fish and the frog. We have the ectoderm, mesoderm, and endoderm. And as the embryo changes, we see that we want to be seeing structures that will appear depending on the interaction between those three layers, okay? So notochord, for instance, that we're going to have it here in the center, is the precursor cells are always placed in the central dorsal portion of the embryo, while the precursors of the neural system lie immediately anterior to it. So that's why we're going to have different fields. But don't worry when I look at that. So to do this, we use different techniques. We use embryos of certain species that are transparent, like the zebrafish. That's what is one of the most used because we can follow that in 3 days. We can see all the process through the translucent egg, okay? Some embryos, however, have opaque cells with a much greater number of cells, so to use pigments become a little more of necessary. Okay, so we're gonna normally use vital dyes because we don't want the dye to kill the embryo, then we don't see where the cells went. Okay, which are simply current that diffuses in the cell, and we can follow their development by following the movement of the layers. Okay, but as cells divide, it becomes more diluted, so at the very end, sometimes it's hard to see where they went. And in some cases, also, we're gonna be using radioactive labels, okay? Or we're gonna be using fluorescent dyes. See, this is all my language coming up together. So that allow us also to incorporate these dyes into the embryo DNA, right? And then we can follow the divisions of those cells. And this is what we see in the screen. So we have a little bit of a dye here. Is the fate map for fluorescent dye. It was placed at the beginning. Here we see that we just continue here with the epithelium of the tissue, and here we have no relation happening. So whatever was in here, they injected the dye in there during late cleavage. And here we see how now this is all part of the formation of the brain, forebrain and midbrain in this embryo. And this is the fate map using whatever they were able to gather here, which part of the embryo is formed. And the dye was only injected 6 hours after fertilization. So we only have a couple of cells in this. And then here we see, oh, okay, this went to the midbrain and this one went to the hindbrain. Darwin also noted that embryos sometimes make structures inappropriate for the adult form, which are later lost. For example, the eyes on the embryonic moles, they have eyes, but they don't use them as an adult. The teeth in embryonic baleen whales, for instance, they have teeth, but then they lose them later. It's just because a lot of these larvae, they need those structures, are essential, but as an adult, they are not. So we have, in this case here, we have the barnacle and we have the shrimp. The embryos are very similar, but the adult form are really completely different, okay? We can also have some homologies. So homologous structures are organs whose underlying similarity arises from they being derived from the common ancestral structure. In this case, this is what we have in the screen, the wing of a bird and the forelimb of a human, okay? They are in the anterior portion, right? Then we also have analogous structures. And the analogous structures are organs whose similarities comes from their performing a common function, not a common ancestor, right? For instance, the wing of a butterfly and the wing of a bird are analogous. They're wings, they help to fly, right? However, we know that a butterfly and a bird are completely different, right? So homologous structure must be compared carefully and must always refer to the level of organization in order to be compared, okay? So a bird and a bat wing are homologous as forelimbs, right? But not as wings. They are analogous as wings, okay? In here, we have the development of the bat and the mouse forelimbs. So they keep the membrane in the bats, but in the rat, we lose it. Even though the field formation of the limb, it follows exactly the same pattern until the membrane is loose in here. So these are great examples of homologous anatomical structures, okay? And okay, I'm gonna leave it there, okay? So you can go grab a coffee, go to the bathroom before your other lecture, okay? And we will resume on Thursday. Just to let you know, this Friday, I said it's not a lab, but I will lecture during that section, okay? And this is just so I can cover some material before we can advance, which means that we're going to finish the course a little earlier in the semester. That I don't think you're going to be upset of that. So we're going to cover the material a little, and I'm going to be using a couple of labs that I didn't program for a lecture, but we're going to use it. I didn't program for a lab, but we're going to use it for a lecture. Okay, so it was my pleasure meeting you guys today, and I'm just going to take this off. And then I need to, this is always my point, what I do here, where I am, what is my mouse? Okay, here is my mouse. Okay, so see you Thursday. Have a great day today, and let's do this together. We can do it together, okay? Bye-bye, guys.
Morning, Dr. Marie. It's Anish here.
Anish, how are you, Anish?
I'm very well. Happy New Year to you. I have a question for you. Are we meeting at 10 a.m. or 1 p.m. on Friday?
For the ones that have the lab at 10, at 10. I asked if everybody could be switched into 1, but I think there are still 3 students registered at 1.
Okay. Because I haven't been able to register for the lab sections yet. Somehow my web advisor won't let me register for any classes. The only class I'm registered for is this one. And I've emailed them and I haven't heard back from them.
Lucky me.
Huh?
Lucky me.
Lucky me. I was able to start the semester with you. All right.
Okay. Yeah, they are behind in registration. I received a couple of emails this morning that students couldn't register, so I sent for 1, but I can send an email to. If you can register to the lab at 1, that will be better for me.
Sounds good. And if I'm able to, if I hear back from them in a day or 2, I'll let you know. If not, then I'll just, if you're able to email on my behalf, I'd appreciate that a lot.
Yeah.
All
Perfect.
Right. Thank you. Have a beautiful week ahead.
Does anybody else have any more question? Luis, Amanda? Annalie?
Oui.
Ok, so je vais juste